Good afternoon, everyone. You all are warmly welcome to today's QAAFI science webinar titled Understanding Nitrogen Efficiency in Beef Cattle, Insight from 7 years of research. So thank you for joining us today. I'm Sampath Disarika, final year PhD student based in QAAFI Gatton campus. So I'm very excited to moderate this webinar today as the topic is very important and highly relevant to many of us. First, I would like to acknowledge the traditional owners and their custodianship of the land which we meet today. On behalf of the traditional owners, I pay respect to their ancestors and their descendants who continue cultural and spiritual connections to country. We recognize their valuable contribution to Australian and global society. So before we begin, there's few housekeeping things, and this webinar is already scheduled for 1 hour from 12 to 1 p.m. And so at the end of this webinar, there will be a question and answer session. You all are welcome to raise your questions. So please use the Q&A tab for post your questions, or you can raise your hand, then we can allow you to talk at the end of the webinar. You all are welcome to ask any of the questions related to the webinar. Right. So I would like to introduce today's speaker. I'm very excited to introduce today's speaker because he's my principal supervisor. So he's an associate professor at QAAFI, University of Queensland, specialising in ruminant nutrition. His research explores the biological mechanisms that derive feed efficiency and maternal performance in tropical cattle, supported by strong industry and scientific partnership. So with experience in Brazil, Australia, and the United States, he brings a global perspective to northern beef systems. He has led multiple major research programs. At present, he's leading QAAFI project, and he published widely and supervised numerous PhD students and master's students, integrating nutrition, psychology, and physiology and genomic tools to improve the cattle productivity. So apart from this academic profile, so I would like to say something about very interesting things about him. At present his ambition is to be a great tennis player. So most of the weekends he's spending practicing tennis. And another interesting thing is he loves ice cream after the dinner. All right, with that very interesting introduction, I would like to invite this wonderful presenter today. Please welcome Dr. Luis Prada Silva for the presentation. Thank you so much.
Thanks, Sam. That was a funny remark you had there. Well, thanks everyone. Thanks for coming. So yeah, today we're gonna do kind of a history on what I've been doing for the past 7 years of research around the nitrogen efficiency in beef cattle. And coming from Brazil, a high rainfall country, the first thing that struck me when I was driving and traveling around northern Queensland was how dry it is, how harsh the environment is for anyone to live there, and the cows are no exception. So I want to start with section 1, the provocation. And behind all the talk today and this line of research, it lies this fundamental proposal in animal nutrition that, look, we're selecting animals, usually feeding them with a high protein diet and maintaining them in a high comfort environment, because that's how they perform the best, and hence they can perform to their genetic ability, right? But at the same time, we expect this genetic to perform in a low protein, harsh environment in Western Australia. And I chose to use this picture with the camel there just to illustrate this point that really these animals need to perform in an environment that demands different physiological mechanisms than this one. So just to put this in simpler terms, the nutrition, the physiological mechanism to make an animal be more efficient in this very benign environment must be very different than the physiological mechanism that allows the animal to be very efficient in this very harsh environment. So that is a fundamental problem that I was faced when I first arrived in Australia. And really, we don't know a lot about how and why animals are efficient in this type of environment. So given that initial provocation, the first thing is, well, cattle, ruminants in general, they have one major advantage over other species, and that is that they can conserve nitrogen. They can preserve the protein in the diet better than other species. If I eat a big steak, there's no way my body can store the extra protein. I'm going to lose this extra protein in the urine. But cattle, not necessarily. They can recycle this back to the rumen and make more microbial protein, new protein to be absorbed in the intestine and go back to the metabolism. So they're able to maintain the nitrogen recycling. Okay, so that's an amazing advantage of cattle. And when we compare the species, you can see that the recycling capacity of cows, steers, sheep, the ruminants, is much greater than ours. We humans, we can recycle some protein back to the intestine, to the large intestine. We can produce microbial protein, but we're not able to absorb that. And carnivores, they do very little of that. So the point is that, okay, we have a species here, cattle, that they're really good at using nitrogen. They are able to recycle. That should give them an advantage. They should be more efficient in using protein. So now that's the paradox. They are not. When you look at the systems and we compare the nitrogen use efficiency, this is a study comparing different production systems, different livestock species in different countries in Europe. And you can see that the average of nitrogen use efficiency is how much of the protein that is being fed ends up on productive tissues, like in the case of beef, the carcass, in the case of milk, the milk, and so on. So only 20 to 22% of the total nitrogen that the animal consumes is actually deposited on the carcass. In the case of beef, in the case of milk, is a little bit better. But you can see that the monogastrics, and especially chicken, they're over 50%. So cattle lose 75 to 80% of the total nitrogen intake. Well, that's hard to reconcile with the fact that these animals are really amazing in preserving nitrogen. So just illustrating this point a little bit better, I'm going to use these slides a couple of times, so I thought it would be good to get familiarized with the concept. Here I have a grazing beef cow, and she's eating during the whole year a diet with about 10% protein. So she's eating about 53 kilograms of nitrogen during the year. From this 53, she's losing 14 kilos on the feces, more or less. She's losing 33 kilograms in the urine. And she's only producing something useful, in the case of a cow, in the calf, in the weaner. And she's going to wean a calf with about 180 kilograms. So really, the only useful part of this nitrogen is being deposited on the calf. So that's 5.4 kilograms of nitrogen on the weaned calf. So that gives us about a 10% of nitrogen use efficiency. So a very low number. So that's the nitrogen efficiency paradox. Like we have cattle here, they're the most efficient species in preserving nitrogen. But when you look at the systems, they are the least efficient in using dietary nitrogen. And behind that, it's the fact that we tend to feed cattle. I showed you the graphs in Europe. We're feeding cattle with high protein diets. We want the animals to perform well. And when cattle are fed with high protein diets, they are not efficient. Okay, so being efficient in low protein diets does not mean being efficient in high protein diets. And the summary of that is that we're not making use of this amazing advantage that these animals have to recycle nitrogen and to produce well in low protein diets. So let me illustrate this point a little bit better. This is a study with dairy cows, and the black line here is increasing the intake. So I'm giving these animals a better diet, and I want to maximize the milk production of these animals. So I increase protein intake in the diet, and I see a linear increase in milk production. Okay, that black dots here, this is the nitrogen that is being excreted in the milk. So I have more milk being produced. The digestibility follows intake. So there's a linear increase in the nitrogen being excreted in the feces. But look what happens with the nitrogen being excreted in the urine. I'm going from 100 grams per day to 300 grams per day. So a 300% increase in nitrogen excretion in the urine to gain a little bit of increase in milk production. So this is actually 10% increase in milk production and 300% increase in nitrogen being excreted in the urine. So I'm going really quickly from an efficient situation to an inefficient situation because we're always chasing production. And then I'm feeding more protein to the animal. So look, the first message of this talk is that, and that I learned when I was studying and thinking about this, is that we're not taking full advantage of the nitrogen recycling cattle. Cattle are inefficient at the system level with low nitrogen use efficiency, high losses in ammonia and nitrous oxide that contributes to the greenhouse effect. And yet, these animals are biologically the most efficient species in using protein. So I thought, well, we need to understand this better. And then we did this review published in 2019, nitrogen recycling and feed efficiency of cattle fed protein restricted diets. And so that helped me to understand the concept of what was going on. And I can illustrate that in 2 slides, but I think you guys already got the point. But let me quantify what's happening here, okay? What is the magnitude of nitrogen recycling? So by changing the protein in the diet from 5% to 20%, which is a reasonable range of protein in the diet that we see in the literature, and each dot on these 2 graphs represent a study. So this is a meta-analysis of studies looking at nitrogen balance. So when I'm feeding cattle with a low protein diet, only 20% of the total nitrogen is being lost in the urine, but this goes up really fast. And when I'm feeding animals with 20% protein in the diet, these animals are losing 60% of that in the urine. So again, quickly, I'm turning an efficient animal into an inefficient one. And if we think about the amount of nitrogen that is coming from the diet, compared with the amount of nitrogen that is being recycled, when you have 100 here, it means that the same amount that is coming from the diet is also coming from nitrogen recycling. So it's a significant amount. So with a diet with 10% protein, the same amount that the animal is receiving from intake is coming from recycling. So it's a massive amount. And we, as nutritionists, we are not able to consider that when formulating diets. We tend to ignore this massive contribution from the animal metabolism to supply the same amount of protein to the rumen and large intestine that is coming from the diet. And for diets below 10%, there's actually more protein or nitrogen going to the rumen from the metabolism than the diet. Okay, so this is a very significant source of nitrogen that should be considered when formulating diets, but it's really hard to predict, and therefore we tend to overfeed protein. And the other thing that is very clear is when we increase digestibility, we increase the efficiency of the animal because we increase recycling. It pulls more nitrogen into the rumen, and also because by increasing digestibility, I decrease the losses on the feces. So nitrogen excretion is highly correlated with diet digestibility and animal performance. And then we arrive at the second message for today is that, look, we don't do a good job in predicting nitrogen recycling. It is very important for the animal. It is a massive amount. It is as important as the diet itself. But yet, we are not able to handle that really well. So we tend to feed high protein diets because we cannot calculate well how much can be supplied by the animal. We tend to feed everything with the diet. And therefore, we're typically overfeeding protein to cattle to maximize performance. Okay? So that's what we learned studying this subject. So the next question was, okay, that's very interesting. Recycling is important, but is there significant individual variation in nitrogen use efficiency? Can we select animals with better nitrogen use efficiency? So that is the second paper. It was published in 2020 when we look at the mechanism of feed efficiency in Bos Indicus steers fed with low or high protein diets. And to start thinking about this individual variation in nitrogen use efficiency, I thought about breeds, okay? We have breed differences. For example, you have these European goats and you have desert adapted goats. So animals that are better adapted to harsh environment. What happens with this mechanism? And what happens is that these tough desert goats, they are much better at recycling nitrogen and lose less nitrogen in the urine when fed similar diets, protein-restricted diets. These European goats, they are not as good as the desert goats to handle these low-protein diets because they're not able to recycle the same amount, and they lose more in the urine, okay? And in cattle, you have some basic studies showing, comparing the Brahma with the Bos Indicus breeds with Bos Taurus breeds, and the conclusion is more or less the same. There's a lot of variation. As anytime you try to compare breeds, there's lots of variation. But in general, Bos Indicus breeds they perform better. They have better digestibility when fed protein-restricted diets, and they have better nitrogen retention when fed low-protein diets. But the ranking is reversed comparing breeds when they're fed higher energy and high protein diets. Okay, so there are indications that's the case. So we put a trial together and we evaluated feed efficiency in 90 Brahma steers, fed low or high protein, both. We did once and then we did the other. And as usual, the range in feed efficiency is significant. Okay, you have a range for more than twofold. You have animals with only 5 kilograms of feed per kilogram of gain, and you have animals with that needs to eat 10 kilograms of feed to gain the same kilogram of gain. So a huge range in feed efficiency, especially in low protein diets. And the ranking of these 90 steers, comparing when they were fed low protein versus high protein, there was a very low agreement in the ranking. So, for example, high efficient animals, only 8 animals were ranked as high efficient in both diets, and only 7 were ranked as low efficient in both diets. There was a big change in the ranking of the animals. And it can be exemplified by these 2 animals here, animal 3142 and animal 3348. When they were fed the normal diet, they both gain about the same, 1.5, 1.6 kilograms per day. 48 gained a little bit less, but was very similar. That was a diet with 14% crude protein and 11 megajoules of metabolized energy. When we decreased the protein to 9%, 3142 melted. It couldn't handle the low protein diet, and the gain dropped to 700 grams a day. And this other guy here, he was able to maintain a pretty good performance. Well, guess what was responsible for this difference? Was the ability of the animal to not lose nitrogen in the urine. The soft steer lost almost 40% of the nitrogen in the urine when fed the low protein diet, and the tough steer lost only 26. So there is a very good correlation between feed efficiency and the nitrogen, the ability of the animal to not lose nitrogen in the urine, so recycle the nitrogen back to the rumen, in our trial with these 90 steers. So, the more efficient animals lost less nitrogen in the urine. Okay, so that means perhaps we can breed for nitrogen use efficiency. You have animals that are really good at recycling nitrogen, and that makes them more efficient to grow in low protein diets. This is data from dairy cows evaluating nitrogen use efficiency. It's a proxy. They're not really measuring efficiency. They're estimating that based on estimated intake and based on estimated milk production. But there is a huge variation. Again, a normal curve of distribution, and in this case, heritability for the trait was 13%. Okay, but look at the range of nitrogen use efficiency in these animals. So there's a large individual variation for nitrogen use efficiency. So if that's the case, that means yes, we can breed for nitrogen use efficiency. However, selection for these traits should consider the amount of protein in the diet. We should probably evaluate the animals on diets that at least don't have excess protein. They need to be restricted in protein values, so the animals are able to demonstrate their ability to recycle nitrogen. So what we did then was, okay, the nitrogen recycling is related to feed efficiency, and what we want to measure is the ability of the animal to not lose nitrogen in the urine. Well, if that is the fact, if that is the physiological factor that makes an animal efficient, we can measure that using nitrogen isotopes, and I will show you how. Okay, so that was our third paper for this presentation, published in 2022, using the natural abundance of nitrogen isotopes to identify cattle with greater efficiency. So let's think about this, okay? I have this animal, and the thing is that nitrogen in the feed, it comes in 2 different forms, and we call that stable isotopes. They stay with that molecular mass forever. They don't lose electrons. They're not radioactive. That's why they're stable. And they can be N15 and they can be N14. And we can differentiate them on a mass spec. Okay? So let's assume an animal is eating a diet and it has 100 units of N15 and 100 units of N14. And it has a digestibility of 50%. So I'm going to analyze the feces and I'm going to find 50 units of N15 and 50 units of N14, the same ratio as the diet. It's just digestibility. What is not digested ends up on the feces. Same ratio between the two isotopes. But the urine is different. The enzymes of the animal, they differentiate between the N15 and the N14. And when we analyze urine, you find almost exclusively nitrogen 14. The animal don't lose the nitrogen 15 in the urine. So that means that if I go now and collect any tissue of the cow, it will have a higher nitrogen 15 ratio than the diet because the animal is accumulating nitrogen 15. Okay? So that gives us a powerful tool because depending on the amount or the proportion that is being lost in the urine, that will change the isotope ratio of the animal tissue. So if we think about a more efficient animal will lose less in the urine. What's going to happen? So this animal has a ratio of 50-20, 50 of N15, 20 of N14. So a more efficient animal will lose less, proportionally less nitrogen in the urine. So it's going to lose less nitrogen 14 in the urine. So then the ratio will increase the N14 on the tissue. So I had an animal that had 50-20, and now I have an animal that have 50-40. So a more efficient animal should have more N14 in the hair or in any tissue and less nitrogen 15. I hope that's clear just by comparing these two animals. That's the hypothesis. If an animal that is more efficient should have less nitrogen 15 and more nitrogen 14. So we went back to our steers that completed the trial, that we ranked the steers for how much nitrogen is being lost in the urine. We looked at the nitrogen 15 on the tail hair, and guess what? It was a very good match. The more efficient animals, they have lower nitrogen 15 on the tail hair. And the low efficient animals have higher nitrogen 15 on the tail hair. So that's the biology. And that, as I said, gives us a very powerful tool to evaluate nitrogen use efficiency in cattle. And we started working with the isotopes on tail hair to evaluate that. And we're not the first ones to do this. This is a group in Europe with dairy cows, and they have a very good correlation between isotopes on animal tissue and milk nitrogen efficiency. So the ability of the animal to use the dietary nitrogen and put that into milk. So very good, well, a decent correlation between the two, okay? Even in dairy cows eating normal diets with normal protein. So the next question was, okay, we did this in steers and worked well. Well, if you think about the big problem we have in the north, which is low reproductive efficiency in breeders, if an animal is able to recycle nitrogen, it should be able to perform better in this harsh environment. So let's look at the cows then. Can we apply this technique to evaluate the more efficient breeders, the more efficient cows? So what we did, we were doing a study at the Birdie Station in Richmond, and we classified the animals during 3 years according to their reproductive efficiency. If they came back lactating, if they came back with a calf, they received a score of 1. If they came back not lactating and they were empty, they received 0. So these animals were classified for 3 years in high efficient or low efficient. We went there, we collect the tail hair from these animals. So this is a retrospective analysis. We collect the tail hair of these animals in May of 2019. And we can go back on the hair because we know how fast the hair grow. And we can go back in the hair and go cutting different slices, and we're going back in time. And what we found with these animals is that during the dry season, the more efficient cows, they had a much lower N15 on tail hair than the low efficient cows. And during the wet season, the difference was less. And if you remember 2019, that's when we had that massive wet event in the north that killed 500,000 cows or 500,000 animals. And that happened around February. And that's when the quality of the grass increased. And that's why you see this increase in N15, because that's also related to the level of protein on the diet. In other words, the animals are eating more protein here, so they're becoming less efficient. They are more efficient during the tough time of the year, so lower protein. So yes, we can evaluate fertility, at least retrospectively, collecting the tail hair. The animals that have better efficiency, they have lower N15 during the dry season. So that's my 4th message to you. Nitrogen isotopes in tail hair can be used to identify animals with better nitrogen use efficiency. Both steers and cows, we found good correlation. And I went to a meeting in Albuquerque in America, and they have a baseball team called the Isotopes. And that was named after an episode of The Simpsons, go figure. So the next step was commercial validation. So we used the QAAFI Alive project to evaluate, okay, we've been doing some retrospective studies, but can we collect tail hair from heifers before they have the first calf? Can we collect the tail hair from heifers, evaluate the nitrogen isotopes, and then evaluate the performance on the subsequent year? So we're collecting hair doing preg testing in year 1, mostly dry, and these heifers were pregnant for the first time. And then we're evaluating the performance on the next year, okay? Calf loss, reconception rate, the weaner weight, and total live weight production. So we've been doing that for the last few years, and these are the samples we collected. We have about 4,500 samples collected so far. So these are 4,500 animals that we know the isotopes on the tail hair, and we know the reproductive performance of these animals. And you can see the clusters are the properties here. So the properties in the NT, for example, they will have a lower N15 value than the properties in central Queensland, because they are eating a lower protein diet by the time we collect the hair. And we've been finding, so a very good relationship with calf loss. Heifers with more nitrogen use efficiency, so lower 15 N on the tail hair, they have a much lower chance of losing the calf than heifers with high values of N15 on the tail hair. And this is all within properties, okay? The properties are a random factor in the model. So this relationship is comparing the animals within the same property across all properties. So each 1 unit, this is per 1,000, by the way, each 1 unit in decreasing N15 on the tail hair, that reduced the odds of losing a calf by 23%. So that's very significant, very interesting result. Why this is happening? It could be that these animals, the heifer is more efficient to use the resource, is able to produce colostrum, deliver milk on the first day of life of the calf, is able to maintain milk production better than the other animals, and that could be related to calf loss, but we're speculating here. We thought there would be a correlation between tail hair and reconception rate. We were expecting one. We have not found one. Okay, the p-values are not significant. We did find an effect with weaning weight. So again, heifers that are more efficient in using nitrogen, they're weaning heavier calves, so related to milk production. And each unit in the tail hair represents about 4.2 kilograms in female calves and 2.5 kilograms in male calves. The weight change of cows was not significant. And when you put both of these numbers together, the weight change of the cow and the weaning weight, you have the total live weight production of the animal. And in this case, was related, so animals with lower N15 on the tail hair. In total, adding the weight change of the cow with the weaning weight of the calf, you have a difference of about 9.6 or 10 kilograms of extra live weight production per unit of decrease in the N15 on the tail hair. Okay, so just putting a dollar value behind this, is it worth breeding for nitrogen use efficiency? Well, it might very well be. If you compare animals with 2 units difference, you can find for a property with 2,500 breeders, you can get an extra $63,000 a year from more calves and heavier calves. All right. The other question was, okay, but can we predict the response to urea supplements? Okay, this question came because people will come to me and say, well, I'm feeding a leak to my animals. I don't have to worry about this protein story. Okay, I'm giving urea, I'm giving the nitrogen to the animals. Why should I be worried about this nitrogen use efficiency? So, Brandon Fraser is a PhD student, and he did a thesis around this. This was published last year, using the tail hair to predict the response to a urea supplement. And we found, again, a decent correlation between the nitrogen in the tail hair of the animals when they were not. So before they were receiving the supplement, we collect the tail hair, and then we gave the supplement and we evaluate the response. And what this graph is showing is that, look, the animals that are inefficient, that they have higher N15, they don't respond well to the supplement. Probably because they are not able, they have a spike in nitrogen when they eat the supplement, and then they lose that on the urine. So they're not able to recycle that and use this supplement as well as these other animals. These other animals, they gain 400 grams per day when fed the supplement. And the inefficient animals, I know there's a lot of noise there, but they gain between 100 and 200, so much less than the efficient ones eating the same amount of supplement. Okay? So yes, we can predict, but the better genetic should be are the animals that give you the better response to the supplement, actually. It's not the other way around. You cannot, well, I don't think you should think of these more efficient animals as animals that don't need the supplement. You should think of these animals as animals that give you better dollar for the supplement that you give them. And okay, but if I'm feeding urea to my animals, can I still collect the tail hair, or is that going to mess up the ranking? Well, during Brandon's study, we found very good correlation between the tail hair that was collected with or without the supplement. Really, the ranking of the animals didn't change. So there was a very decent correlation in the N15 values for the hair collected with or without the urea supplement. So I don't think it's a problem. I think even when the animals are fed with a urea supplement, you can still collect the animals. The caveat here is that for these animals, we were feeding the same amount of supplement. So in your property, you might have animals that are eating more supplement and animals that are eating less, and that could probably change or will change the tail hair isotopes. Right. It is all interesting, but what's the heritability of this trait? Well, that's when a master student, Morteza, he worked with about 500 steers, so not a huge number. We evaluated these steers. We had known the genetic of these animals, and we were able to calculate the heritability as 43%. So again, it's not a great number of animals. The variation, the standard deviation of the heritability is not small, but it is a significant heritability of 43% for the tail hair. And I think this high heritability comes from the fact that it's a very precise measurement. We're not estimating anything here. We're using mass spec to monitor, to quantify the N15 on tail hair, and that gives us a very good heritability. So when we compare 43% with other traits that are used in cattle, you can see it's a high value, so it's very useful. All righty, so that's the story so far. What's next? Well, we think we began the hunt for the nitrogen use efficiency gene, and we can give a first step by combining this phenotype of the 6,000 cows part of the QAAFI Alive project. When we finish analyzing the last few cows, we have the genotype of these 6,000 animals done in partnership with Weatherby Scientific, and we can match this and look for DNA markers for nitrogen use efficiency in cattle. Of course, this nitrogen use efficiency is being estimated by the isotopes on the tail hair. So this will give us a very good first step in finding the nitrogen use efficiency gene or genes, and being able to convert all of this knowledge into a GBV for nitrogen efficiency. The other area that I think it's really interesting is the relationship between nitrogen use efficiency and heat stress. So Brandon, one chapter of his thesis was looking at animals that went into the climate control chambers and they were exposed to a very high heat stress event. We're calling hot here. What happened to the nitrogen metabolism? So it's the usual response. The intake goes down. The animals breathe faster. They drink more water, and they are much less efficient in using nitrogen. So if they are less efficient, you expect to see an increase in the N15 on the tail hair, and that's exactly what Brandon found. And then this goes back after some time when the animal is normalized. So we can see the heat stress event happening on the tail hair because it changes the nitrogen balance of the animal. And look, the resilience to heat stress is probably related to the nitrogen use efficiency. These are some classic data from John Vercoe. The animals that are more efficient, are more resilient to heat stress, they lose less nitrogen in the urine. They lose less water in the urine, and nitrogen and water, they both go together in the urine. So, and there are other indications that, look, that's a good research question. Are cattle with lower N15 on tail hair, are they also more resilient to heat stress? And can genetics and nutrition interact to improve his stress resilience. So look, these are definitely an area that I want to pursue. So at the end of this, I think we made a very good progress into looking at these animals in an early age, in being able to classify in good, medium, bad. These animals I don't want to breed. These animals I don't not. It's not worth supplementing them based on this what we learn on the nitrogen metabolism and the tail here. Okay, Sam, I'm gonna pass this back to you so we can run the questions and answer section.
Thanks so much, Luis, for your wonderful presentation, and that was really interesting to hear. And thanks for sharing what you have explored throughout the 7 years your research career. And still, the question and answer button is open for you to post your questions. So before I go to the question and answer tab, I have a general question to you, Luis. Like, if you think about the producer's perspective, how we can transfer these findings to this extensive Northern Australian beef cattle system, especially that is dominated by pasture, and especially that is the nitrogen intake is driven by dry season supplementation.
Yeah, look, I think when I talk to producer about this, I think the first point that I really would like the industry to consider is that we need to think about how we select genetics for the North. And I know a lot of people are doing that, and but I think there are better ways to improve the program that we have to evaluate cattle for the North and to consider the efficiency. Because if we're not considering that, we're losing a massive advantage to improve the herd. So that's the first thing that I think is important for the producers. And the other one is, look, if we have a way to identify the more efficient animals, or if we have a way to identify the least efficient animals and remove those from the herd, that's already a great advantage for the producers, because they don't need to maintain the less efficient animals on the property using the resources.
Thanks so much, Luis. That's a nice clarification. Yeah. So we got a lot of questions in our question and answer tab. I want to jump into one question. Do animals on high protein diet consume less total biomass? And if so, what impact does that have on weight gain? Shall I repeat it, Luis?
No, that's okay. No, no, they don't consume less total biomass. They eat more. When you increase the protein on the diet, they usually have a higher intake, and that's associated with a better weight gain. So they now the animals on better protein, because you need to understand how you change the protein on the diet as well. But even if you're just adding some urea for a diet that is lacking urea, you see an increase in intake.
Okay. I think you got a satisfactory answer for that question. And one more, nitrogen efficiency is more likely to relate to the genetic selection, or the dietary nitrogen source could change this efficiency.
No, the nitrogen source definitely changed the efficiency. If we use non-protein nitrogen, such as urea, you find a lower efficiency because the animals will lose that. And there are several studies showing that if you add casein to the abomasum, for example, that increases the efficiency of the animal. So if you give true amino acids, bypassing the rumen will give you a more efficiency, a better efficiency. So now the source of nitrogen, the source of protein is an important part. Genetic selection that's the point. Like, we don't know because we have not been selecting for that. We may have been indirectly selecting for nitrogen use efficiency when we select for cows that are able to conceive every year. But, selecting on females, it's it's a very inefficient way to spread the genetics for the herd. You need to select on the bulls.
Yeah. Thank you, Luis. And this is one more very interesting question. Would this isotope concept be applicable in wool?
Yes, and there are several papers with sheep where, in case of sheep, they're looking more of the provenance. They're trying to use the isotopes on the wool to evaluate what type of diet the animal was eating. So there are different uses for isotope. But yeah, definitely, it will be registered on the wool of the sheep, or it is registered on the wool of the sheep.
All right. Yeah, I think Daniel, you got a good answer for that one. Another question, I just wonder if adding the rumen protective amino acids could change the nitrogen efficiency.
It could. As I said, there are evidence that providing protein post-rumen will increase the efficiency of the animal. And funny enough, it increases the recycle of nitrogen back to the rumen, which is, there might be reasons for that on the enzymes, on the urea cycle that we can, glutamate. But the fact is that, yeah, if you feed amino acids post-rumen, you usually see a better efficiency.
All right. Yeah. So yeah, still we have some more time to discuss more questions. So thank you
We
for
have 5 more
posting.
minutes.
Yeah, 5 more minutes we have.
Yeah.
Another, yeah, another interesting question. After heat stress, nitrogen efficiency recalibrated, but not back to where it was before. Does it go back to eventually?
Yeah, on that study, the recovery phase was just 3 weeks. So probably the animals were still experiencing some side effects of the heat stress. So we don't know. It would be the answer, but I would put a lot of money to say yes, the nitrogen efficiency will go back to where it was before.
Okay, thank you, Luis. And did you also explore the different cattle breeds and nitrogen efficiency? Maybe
No,
between
no.
breeds?
There's a lot of interest to do that. The breed associations, several of them came to talk to me because they want to see if their animals are more efficient than the others. And I'm sure there will be differences. I'm a little reluctant to go into trials comparing breeds because it's not an easy task. Like, you're gonna select how many animals from each breed to say that this breed is better than the other one. And they need to be the important thing about all of these I'm talking about on the nitrogen isotopes is that this only makes sense if the animals are in the same diet in the same paddock. Okay, you saw that the clusters of properties on the calf alive, so the signature is very much defined by the diet that the animal is eating at that moment. So if you want to compare animals, they need to be eating the same thing at the same time. And comparing breeds, there's a lot of variation within breed. So it's a hard one to get into. I'm not sure I will.
Thank
I'm
you,
more
Luis.
interested to comparing the individual variation in feed
Okay.
in nitrogen efficiency.
Yeah, well, all right. One more questions. Do ruminant microorganisms also discriminate against N15? Yeah.
Yeah, they do. That's part of where this discrimination of N15, N14 in the animal comes from. Is from the bacteria that they differentiate the 15 and 14. So yeah, that is definitely part of why we see the N15 accumulating in animal tissues.
Yeah. And the next question is related to genetics, I think. Then that's about, do you think at some point it can be introduced as a new trait in a selection genetic strategy or selection genetic strategies?
Yeah, I believe so. So what we did, after we gather all this information, we contract a consultancy company to ask the producers, okay, would you pay for this test? And the conclusion of that work was that, yeah, most producers, they are interested, they see the value, they will pay for that. Not a lot though, of course. So I think the most efficient way to introduce this into the genetics in the north would be through GBV, through the normal genetic evaluation. I mean, to do that, we probably need DNA markers. Like we could just analyze the tail hair in a lab, but there's no commercial lab doing that. And I think I haven't felt the appetite for investors to put money into a lab to evaluate the tail hair. But I could be wrong. It could be that's the best way to go is to just stay with the tail hair isotope. But for now, I think the best for us to do in the research is to see if we can find DNA markers associated with this trait.
Yeah, thank you, Luis, for that clarification. I think we can go for the last question. We got a lot of questions, but the time constraint, we have to go on. Is there a relationship between nitrogen use efficiency and methane emissions? That could be the last question.
We haven't measured that, but there should be an indirect one on methane intensity. I don't think there will be on emissions, but because the animal have a better feed efficiency, and that has a direct relationship with methane emissions, then you're going to find this indirect relationship between nitrogen use efficiency and methane emissions.
Well, thank you, Luis, for clarifications and answers for all the questions, and thank you for conducting this webinar. And we would like to conclude this webinar for today. And we know that you have a lot of questions. Please, if you can communicate with Luis for these questions. And we would like to share this the code for registering for the seminar invitation list. If you haven't registered yet, please scan this QR code for registering. And thank you very much for everyone for your company. We'd like to wish you a wonderful afternoon. Thank you so much.
Thanks, Sam.